Well, welcome back, everybody. I'm very, very happy to be here. I'm David Ewing Duncan, and I'm a journalist and science writer and author. And I am very, very pleased today to welcome Craig Venter, who has been here before, as have I. In fact, Craig and I have spoken before for one of the fireside chats. Craig is the founder, chairman, and CEO of the J. Craig Venter Institute, and has been one of the great pioneers of our time, really, in biology, genetics. And we're going to talk today about something that some of you may not be as aware that he's done as some other accomplishments that he's had. It's about what we're calling it cataloging Earth, mapping the planet's microbiome. And you think maybe of the microbiome of the human gut, which I'm sure many of you are aware of. And there's been a lot of research, interesting things done on that in recent years. But there's also something called the microbiome of the planet. And this is environmental microbiology. And that's what we're going to talk about today, because the whole span, in a way, of Craig's career, or much of it anyway, has been in this realm. And some of the other things he's done, like sequencing the human genome, the synthetic biology that he's done, has been an outgrowth of his work on trying to figure out what's going on in this invisible world of microbes. And it's an extraordinary world, and I've gotten to view it much more closely than many people in the last 2 or 3 years, because Craig and I have been writing a book together about this, which will come out next year. And it's called The Secret Life of Earth's Smallest Creatures. And so let me jump into this, and we're going to have a little conversation here and talk about the microbiome of the planet, how it relates to some of Craig's other work, and some of the adventures along the way, because as many of you know who know Craig, he doesn't just sit in the lab all day and churn out stuff. He gets out there and gets his hands in it. And this is a case where it's actually an adventure story. And it's a science story, adventure story. It's kind of fascinating. And I did want to add that a lot of the issues here in this conference have been about big things and very important things. But this is about something very, very small. But it's also equally important. So, Craig, let me just start out with helping people that don't know that much about the microbiome of the planet. If you can just kind of describe what it is and why it's important.
Well, we're a planet of microbes. We see large animals because of our visual acuity. But life started in the microbial world, and if we were all wiped out, the microbes would continue to inhabit Earth. It'll be interesting to see if some show up on Mars, because we exchange about 100 kilograms of material between Earth and Mars each year. So if there were microbes on Mars, we have them on Earth and vice versa. So it's a question of some of the deep water reservoirs underneath the surface on Mars, if they contain some ancient microbes that traveled from Earth. But it's you can't take up a teaspoon of soil without getting millions of microbes. Every milliliter of ocean water has 1 million bacteria and 10 million viruses. So the collection of all that is, I guess, what we're calling the Earth biome.
Right, and we're talking too, just to be specific, about bacteria, viruses, fungi, small, tiny, little microscopic animals. And so this is a vast, vast realm. And as I said, there are more microbes than there are stars in the universe. And Larry Smarr, who's an important figure here at FIRE, was actually involved with this project at a certain point. And is in the book. And he, as an astrophysicist, actually got interested in the microbiome and his own microbiome of his gut, which many of you are familiar with, his project to figure out what's going on inside of his own body. And as an astrophysicist, he determined that there are far more microbes than stars. So why isn't he looking at microbes as much as he's looking at stars? So that's the universe that we're talking about. And I might add that it also has enormous influence on this planet. In fact, it's sort of a case to be made, and Craig, maybe you can help with this, that this is really planet microbe, it's not planet human. I mean, they really run things.
Well, the microbiome is the tens of millions of microbes that inhabit our gut, our skin. You know, if you killed all your microbes, your weight would go down 2 or 3 pounds. So we are a giant microbe environment ourselves. And as I said earlier, just every environment on the planet is inhabited with microbes. We have them in the atmosphere floating around. When the pristine lakes in Antarctica were cracked open and exposed, we actually found, and one of our team members was down there doing that, extremely dense populations of ancient microbes that hadn't seen sunlight for a very long time. So you can't find an environment that is not completely microbial driven, but that wasn't the view not so long ago. Our view of the microbial world, you know, scientists aren't necessarily the most brilliant population on the planet. We as a population tend to think extremely narrowly. So the microbial world was based on what could be isolated and grown in culture, which is probably about 1/10,000,000th of the microbes on the planet. So our view of the microbial world, our view of the number of microbes in the ocean, was based entirely on what could be isolated and grown in the laboratory and characterized. And what changed that was an experiment that started back in 1995. I decided that we could use a new mathematical algorithm to assemble genomes. And with sequencing, we break the DNA down into little pieces. You sequence about 500 letters at a time. And so to reconstruct a genome, you have to put all those pieces together. We developed some good algorithms that do that. And that turned microbial sequencing projects from things costing tens of millions and decades to relatively short periods of time. And because of that, Haemophilus influenzae, what we chose to sequence, ended up being the first organism on this planet to have its genome deciphered. The question was, you know, how powerful were these algorithms? And we found out, because we were sequencing pathogens of every type and nature, and we got a pathogen sample from a very sick patient in Norway, and it was supposed to be an infection of strep pneumococcus. And the nice thing about the algorithms, they don't make any assumptions. We just know that there's DNA there, we sequence it, and then out of the assembly, you find out what was there. It turns out, instead of being one microbe, it assembled into 2 independent microbes. And that's where I realized for the first time that every species on this planet has its own mathematical solution for its genome. I think for a while, I was the only one that believed that. I convinced the head of biology at the DOE, Ari Petrinos, to fund a project of taking a sample of seawater from the ocean and doing this shotgun sequencing on everything we could filter out of 200 liters of seawater. The scientific community was certain that this would fail miserably and we would not be able to make any sense out of anything. Fortunately, the DOE can override reviewers' comments, and he funded it anyway. And our first project, just from 200 liters of seawater, we discovered over 1 million new genes and uncountable number of new species. And so that's what that became the start of what we ended up calling the Sorcerer Expedition. As a sailor, you know, I wanted to sail my own boat around the globe. Most sailors fantasize about doing that. Less than 1/1,000,000th of them actually do it. But I found a way, and it pissed off a huge number of people because I found a way to sail my own boat around the ocean and do great science at the same time. And the notion was, you know, we could have kept sampling in Bermuda, but I thought maybe in different parts of the ocean, there'd be different microbes that were different abundances, and we would get a consensus built up much faster by sampling around the globe. So we decided to equip my yacht and for science, and took samples every 200 miles and found that they were over 80% different every 200 miles. And that's where we'd be able to think of the, you know, what you've been calling the planet biome, that it was much more complex by several orders of magnitude than anybody ever imagined, and that the oceans weren't a giant homogeneous soup. You know, maybe they're different a mile away. Now, when, you know, we look at the satellite images, you can see the algae blooms, you know, they have very sharp borders. So in some cases, you could measure 500 feet away and have a totally different set of microbes.
Yeah. Well, just what Craig just described is an incredible trajectory. I mean, he, you know, became well known in the early 90s for really wanting to speed up the technology, you know, algorithms around sequencing, you know, which was very slow and cumbersome at the time, and was moving at such a slow pace. Who knows when the human genome would have been sequenced and, you know, other organisms sequenced. So Craig was, you know, is very familiar with speed. He races boats. He loves that, and he translated that into this trajectory that started in the mid 90s when he sequenced the first organism, which was a bacteria, as he mentioned, and in a really extraordinarily fast pace, got us, you know, within 4 or 5 years, was sequencing the human genome using that same technology. But it was really the bacteria. Correct me if I'm wrong here, Craig. That you demonstrated the process that would then work on the human genome and other organisms, and then you know, while that was still going on, you translated that into taking what you once called, I think in Wired magazine, you were going to go out and sequence the planet, and, you know, obviously that's not possible at the moment, but the idea of going global was, you know, an extraordinary, kind of almost revolutionary way of thinking about this, because scientists up to that point had thought, you know, I'm going to spend my career in an estuary, you know, studying every little bacteria and microbe there. And you said, let's do the ocean. And so you hopped in your boat and you were off and running. And let's talk about that for a minute. You started in Bermuda with the Sargasso Sea, and you blew everybody away with what was there. Explain what you found that was so startling.
Well,
back in 2004.
Yeah, the discoveries of organisms in the Sargasso Sea, which is the area surrounding Bermuda and the southern Atlantic Ocean, based on the Sargasso Sea grass that grows there. The discoveries were numbered individually, and they're up to SAR 11. So SAR stood for Sargasso Sea, and they were up to the 11th microbe discovered there that they could culture. So in one experiment, one sequencing run, we discovered close to 1 million microbes. And that's not what anybody expected. And it's funny when we make these quantitative leaps, it actually makes some people very angry. When we did this first with human genes, I got hundreds of angry letters and emails of all the doctoral theses I ruined, because people were going to take 10 years to isolate that one gene. And the same thing with oceanographers. You know, they wanted to continue their slow count of discovering one microbe at a time, and in attributing vast characteristics to it. So, you know, instead of leaping forward in science, as you know, we sort of went back to the way science was done in the 1800s. So everybody thinks of Darwin. Darwin wasn't actually on a scientific expedition. He was on a survey ship, but these ships always took along a naturalist to help characterize things. But the Challenger expedition was the first actual oceanographic expedition. And it was based on the same naivety. Scientists were absolutely sure life would not exist below a certain depth in the ocean.
This is in the 1870s, right?
Yes, 1870s. So, you know, we come up with these absolutes. In the human genome, everybody heard about junk DNA because genes actually occupy such a small amount of the genome. And I mean, they're actually just, we're expressing our ignorance in the most profound way, because we don't know what something is. It's junk or it's useless. But it turns out what the Challenger did, they stopped every 200 miles. So how does 200 miles fit in? Those of you who are sailors know that's roughly the distance a good sailing vessel can go in 24 hours. So taking samples basically once a day on a voyage. The Challenger, instead of filtering seawater, dropped a dredge to the bottom of the ocean and scooped up and pulled up, and they discovered new life everywhere they dropped the dredge. And this sort of blew people's mind equally at the time. And it's, what is it, a 20-volume set describing mostly macro species that they discovered, clams, shrimp, all kinds of sea creatures. So we were mimicking, if anything, the Challenger expedition from the 1870s of sailing around the world, only this time just taking seawater samples. Now, the big difference between the 1870s and also what Darwin did and what we could do today is every country owns all the life up to 100 miles off their coast. So, as I said, you know, there's roughly a 1-knot current going across the Pacific. Microbes, fish, whatever, as they get into within 100 miles of the Marquesia Islands, they go from being belonging to planet Earth to becoming French genetic heritage. And so you have to have permission to even take a sample of seawater. So we had to work through the State Department and with militaries to actually get permits to sample in every country when we were within 100 miles of the coast. And that led to some adventures. We were arrested 3 different times. And the boat was threatened to be sunk by the French Navy. The US Navy had to intervene and contacted the French Navy and told them that we were a US research vessel, not just a private yacht, and we were under the protection of the US Navy. And it took that to get them to back off. But there's so many discoveries, so many directions. You're the host. Tell me, steer us where you want to go.
Well, I did want to sort of add that we're going to get to in a minute the here and now. This is a little bit of how this story unfolded in the history of this rather extraordinary sort of scientific journey. But as Craig said earlier, microbes are everywhere. They are in the air, in the atmosphere. They actually are one of the reasons it rains. The ocean is a gigantic pump that absorbs carbon and produces oxygen, on and on. And in fact, microbes are really why we have an oxygen atmosphere. So if you're kind of wondering where we're headed here, we had Craig and other scientists had to figure out a way to understand this planet microbe before we could get to where we are now, which is trying to understand what humans are doing to it. It is another aspect of human activities affecting the planet, and it's a little known facet of that, but it's a very important part of it. But going back in time a little bit, Craig, you took this global expedition, this circumnavigation, 2004 to 2006, and then there was another expedition that went to Europe, the different seas there, Black Sea, Baltic, et cetera. The boat, I forget the total mileage, but it was some extraordinary number.
65,000 miles at sea.
Yeah. So what were you looking for, and what did you find in terms of microbes?
Well, it was like last century science. I was just going out to look to see what was there. That's why it wasn't so popular, because our government agencies have turned into, you have to have a hypothesis that you're trying to prove or disprove to, quote, be doing science. We were on what was called derogatorily a fishing expedition. Fortunately, we did do a lot of fishing. We always had lines in the water trailing behind the boat. And I would say at least half the time we had fresh ahi, tuna, and fresh sushi every night to consume. So it was a fishing expedition, but we didn't know what we were going to find with the microbes and with the viruses. And there's so many stories within stories. So I mentioned there's 10 million viruses. So there's 10 viruses for every microbe in the ocean. And these viruses or phage actually can help control the microbial populations. But viruses, we think of things, particularly with the last couple of years, as things that cause pandemics and harm to humans. Viruses actually don't want to kill their host. And one of the most fascinating discoveries one of our scientists made, Shannon Williamson, was that for the photosynthetic microbes, the viruses or phage that control their growth actually upgrade the genetic software each time they infect them. So they improve and correct errors in the genes for photosynthesis, keeping this microbial population healthy and functioning. So, you know, in a bizarre philosophical way, the viruses are sort of in charge keeping their host alive and renewing their genetic software so they can always reproduce. So, you know, it makes you wonder who's really in charge on the planet. You know, it may be the viruses after all, not the microbes or the macro species, but the notion that the viruses actually improve the viability and the software within each species in the ocean, I think is one of the most profound discoveries.
Yeah, I mean, I was blown away by that when I read Shannon Williamson's paper. And let's see, we humans we have about 8% of our genome comes from viruses that go back some of the millions of years, that organisms that we evolved from and we ourselves, you know, have accumulated. Is that about right? I mean,
I think that's approximately correct. You know, each of our genomes contains not only our history, but the history of our species. And that's how we can track from early Southern Africa to today, certain parts of genes that we have, and with the other populations of humans, the Desnovians and others, we can track these and we can track portions of our genomes that contain those. Likewise, we can track the microbes and how things evolve. So, for example, eukaryotes, we're a member of eukaryotes. It means our cells have a nucleus, was actually a fusion of two bacteria into each other. And the same thing with all plants and photosynthetic organisms. A photosynthetic bacteria got incorporated into another bacteria and became the photosynthetic core of what we now know as eukaryotic plant cells, and became plant life. So the accidental merger of cells going together, you know, there's been big battles in scientific communities, you know, how many chromosomes different genomes had, and how they derived. And that's the nice thing about taking an algorithmic approach. As I said, there's no a priori assumptions. There was a big battle with cholera about how many chromosomes it was, and Carl Woese argued that it was identical to E. coli because the 16S RNA gene was basically the same as E. coli. And what we found on sequencing the cholera genome is it had 2 chromosomes and a very large plasmid, 100,000 base pairs long, that like a mini chromosome. So life looks very different when you have a naive approach that's not based on any preconceived notions. And I think that's how we made so many discoveries around the globe and discovered so many differences and yet so many overall principles. The genes reflect the chemistry of the water around them, or maybe the water reflects the chemistry of the genes of the microbes. In the Eastern Pacific, there's differences in chloride levels, and the genetics of the microbes have totally different chloride metabolism than they do in the Western Pacific. But we also found that temperature affects everything, affects the amount of CO2 the ocean can absorb. It affects whether microbes live or die. And I don't know how many of the audience have been to the Seychelles. The Seychelles have some of the most beautiful white sandy beaches. But it turns out that's a terrible tragedy due to a 1 degree increase in ocean temperatures. A key microbe that kept the coral cells alive couldn't survive with that increased 1 degree temperature. And so it died, and that left the coral to so-called bleach out and die. And the destruction of the coral reefs is how we end up with the white sandy beaches. But we're losing key parts of the future viability of the ocean. You know, people are talking now about things have gone so far with CO2. We have to do massive global CO2 sequestration. But the problem is, if we take 1 million tons of CO2 out of the atmosphere, we actually have to wait for re-equilibration of the CO2 in the ocean, which would get released back into the atmosphere. So it's not easy just to remove it from the planet. We're part of a giant equilibrium system that's going to take massive changes to get it to survive.
Yeah, this is the last chapter of the book is called The Microbial Inconvenient Truth, which is riffing a little bit off of Al Gore's Inconvenient Truth. And that refers to, in part, what Craig is saying, that as we, pump carbon into the atmosphere, we have nitrogen and phosphates dumping into the oceans from the rivers that run off from fertilizers, different pollutants around the planet. It is changing the microbiome of the planet. The analogy is, if you eat junk food, you're going to change the composition of the microbes in your gut, and you probably will get sick. And the difference is, though, as Craig has said earlier, we can change the composition of the microbiome of the planet by these pollutants and other human activity. They will be okay. The question is whether we humans and the ecosystem that we thrive in will be okay. And so this is something that we're going to be talking about, as the book comes out, and already are starting to talk about, because it's not widely known outside of oceanography, microbiology, that there is this whole hidden sort of story about the impact of human activity on the planet. And this, of course, wouldn't have been possible without this story we're telling, which is why we went through that. And it's also an exciting and fascinating story. But that's really in the here and now. And what Craig's Institute, many others, in fact, there's been an explosion of interest in this field in the last 10 years since, or 15 years since the initial expeditions of Sorcerer II. And this is what's really the pressing issue that we wanted to bring you all and talk about. And Craig, maybe you can even get into a little more detail about just how important microbes are in the ocean. I mean, we can talk about the air, we can talk about the soil, but this pump you were talking about a little bit, but the microbes in the ocean sequester about 30 or 40% of the carbon in the atmosphere. And that's under stress right now, for instance. Maybe you give us a little, sense of what's going on and why we might be worried about that.
Well, the photosynthetic organisms in the ocean, these small photosynthetic bacteria, produce at least 40% of the oxygen that we breathe in our atmosphere. In fact, it was changing from anaerobes to organisms that actually produced oxygen that changed the history of life on the planet. And with the evolution of plants on the Earth, coupled with all the oxygen coming out of the ocean, that's what created our oxygen environment and atmosphere. that allowed us, for example, to be here. There's now huge parts of the ocean. There's a huge dead zone off of Oregon. There's now one off of California. These are cropping up all over the planet where there's no oxygen left in these zones. So, you'll see the dead fish floating to the surface. Normal life cannot survive in these, but anaerobes can. But the changing the amount of oxygen and the organisms that are there obviously change the amount of CO2 that can be sequestered. The microbes are, even though they produce oxygen, they're also a very key part of the food chain. These algae blooms sometimes produce toxins that work their way up the food chain. For example, those of you who've been out sailing around the world know that you never fish inside the outer reef on islands in the Pacific because the large fish that you catch, whether it be tuna or barracuda, by being at the top of the food chain, have consumed these microbes that contain toxins, and they make humans quite sick when you eat them. So that only happened once on the expedition. We thought we were far enough close to the edge of the outer reef that the fish would probably be okay, but the crew got quite sick, and one person passed out from eating contaminated fish. But we have toxins produced in these algae blooms off our coast that can end up in food we eat and cause bizarre behavior in sea mammals, bird life, etc. But microbes are also a key part of the geopolitical arena. The reason the French, for example, arrested us when we entered the Marquesas, they were afraid that we were going to go over and sample from the waters surrounding where they did all their nuclear tests. And they were afraid that we would show massive mutations in the organisms that we found there. So that's why they threatened to sink us if we went over there to sample, even though our permits, we didn't even ask for those. The British arrested us and threatened to impound the boat, took our passports, held us at gunpoint in the Indian Ocean because most people have heard of Diego Garcia, where we have a B-52 base in the Indian Ocean. That's part of a series of islands where the British removed all the natives and leased it, Diego Garcia, to the US for the B-52 base. There's this one extremely beautiful island with probably the most beautiful coral reef and atoll on the planet that cruising sailors are allowed to stop at. We notified the State Department, it's halfway across the Indian Ocean, that we're going to stop there for a night or two to rest from having a rough trip in the ocean. But the British were looking for us and came and boarded us and arrested us. And what they were worried about was all the attention that I was getting and the Sorcerer expedition was getting would bring to those islands, and the fact that they lost in world court 3 or 4 times to let the natives return to the islands, and the British still haven't done that. So geopolitics enters into some of the most bizarre ways to this, and human activity. So in Australia, we were boarded by a heavily armed troop of police while we were in Brisbane, because one of our crew members was dating a woman whose ex-boyfriend got very upset and told the police that we had a meth lab on board our vessel. And so they wanted to board it to inspect it to see if we had a meth lab. I negotiated with them because I didn't have to let them board, being an U.S. research vessel. They didn't have a warrant, but I negotiated with them that if they would take off their combat boots and keep their weapons holstered, that they could come aboard, and if they wanted something opened, a crew member would open it for them. But as soon as they got below, they realized they were on a fairly luxurious yacht with scientific equipment, and it didn't smell like a meth lab. So science has some interesting prospects, whether it be from disgruntled lovers or from disgruntled countries.
I was wondering if you're going to tell that story, but so we do have 4 or 5 minutes left here in our discussion period, and there are some questions already popping up in the chat. I would love for anyone else who has questions to start thinking about them, putting them in the chat. And I believe Evan is going to help moderate that part. But in the last 3 or 4 minutes here, Craig, I did want to give you a shout out that the New Yorker has a story, I believe it's this week, on synthetic biology, which is another area which we barely touched on here, but that you're associated with in a very profound way, having sequenced or having actually created the first artificial organism, which then worked. Or at least creating the genome in a artificial organism. And this article this week describes the projects that came out of that, and it's sort of reshaped how synthetic biology is being practiced these days. But tell us how that syncs with this whole story. I mean, we've got the microbes, we've got the sequencing, and where does synthetic biology come in?
So when we sequenced the first microbe, we actually did 2 different genomes in 1995, thinking that comparative genomics would allow us to understand what the genome was telling us. And there were so many unknown genes and new factors in the genome. As we sequenced more and more species, we could solve some things. But we actually, took a hint from Richard Feynman, who said that something to the effect that to understand it, you have to build it. And so we decided, we being Ham Smith, Clyde Hutchison, and myself, that the only way to really understand these microorganisms and how their genes contributed to them being alive was to synthetically make a chromosome, first just to see if that was possible, and then to try and design a chromosome based on our knowledge and see if we could boot it up and make a synthetic cell. We achieved the first step in 2010, and that made international news and received comments from President Obama and the Pope simultaneously. President Obama asked his new bioethics committee to look at what we were doing and report on how dangerous it was to the world. And the Pope said, Craig does not create life, he just changed one of life's motors. So different views from the beginning, but it took us 10 years to design and build a chromosome. And the reason it took so long is in our final construct, about a quarter of the genes are still of unknown function. So biologists think they know basically all of biology. We proved that we're missing at least a third or more of fundamental understanding of biology. And so we had to do random replacement experiments until we could get viable cells. So nobody else has yet made a synthetic cell because it's a long, arduous process, but there's some very exciting technology coming now that makes writing the genetic code almost as fast as reading it. And that will, I think, make it possible to make thousands of synthetic cells to start doing what we would like them to do.
Right. And obviously that has all kinds of implications for science, for commerce. You mentioned ethics. I mean, David Brin just asked, you know, can we program these little guys to, you know, die if they leave the lab? And that, in fact, was something that was brought up long ago in the 70s with, I think Sidney Brenner said during the debate over recombinant DNA, when everybody was worried about, you know, putting genes into bacteria, you get loose in the environment and kill us all. And so that's been a discussion that's been around for quite a while. But anyway, I don't know. Evan, do you want to jump in here with questions?
Sure, yeah. Really briefly, Craig, Rennie was trying to figure out the paper that you mentioned about viruses in the ocean. And he posted a link to one with Sikowski, Munsell, Hyatt, Womack, Williamson. Does that sound right? It's called
Yeah, it's 2007, and it's in PLOS.
Oh, okay.
Yeah, we can get that link out. But there's a special collection that PLOS put out in 2007, 2008 about the Craig's the whole expedition, lots of different aspects of it. And I forget what it's called. It's Ocean Microbiome Collection or something like that. Yeah. And so it's part of that.
Sorcerer Sailing in the Ocean is on the cover.
Yeah. Okay. Yeah, thank you. So Barrett was asking, Barrett, do you want to ask your question? She's not there. Barrett was asking, when you were talking about genetic diversity and some of the, you know, earlier in the conversation in the ocean and threats to the ocean based on temperature, et cetera, she said, how does this understanding of genetic diversity of the oceans play into our understanding of ocean health? For example, does that make the world's oceans more or less resilient to the potentially devastating impacts of deep sea mining? And as a follow-on, are you involved at all in those kinds of conversations at the UN?
We'll answer the last question. That last part is no, I'm not. But I think by understanding the life cycle and the diversity of all these microbes and their role in producing carbon, eating carbon, there's ways that this has been looked at. We were asked by BP to look for microbes deep in the earth that produce methane from CO2. Now, they wanted to use the methane, you know, as natural gas, but methane has about 20 times the negative potential as the CO2 molecule does. So there's huge reservoirs of methane in the ocean that we'd actually like to see converted to something else. There's huge amounts in the deep ocean that, if it was all released, would basically suffocate mammals. So the ocean environment and changing it. I mean, this is the fire conference. We're playing with fire if we really try and geoengineer the oceans and the atmosphere. So even CO2 sequestration, while it sounds logical, as I said earlier, it shifts the equilibrium in the oceans, and we're playing on a global scale with things that we still don't truly understand. It's clear humans are genetically miserable at preventative medicine. We're struggling this in the actual practice of medicine, where we can discover diseases in people through the genome and through new imaging techniques. And the medical establishment is pretty much against doing this early detection because all their money is made by people coming in with symptoms. We see the symptoms in the environment, but we don't want to change what we do to prevent things. So geoengineering may be our only choice, but it's like playing Russian roulette with 5 bullets in the chamber instead of just 1.
Yeah, and somebody asked about the equilibrium of the planet, and that was a wonderful description of what happens when you mess with it. But yeah, yes, I mean, there is a rather extraordinary equilibrium that the microbiome of the planet has struck. And again, that's supporting the ecosystem that we live in right now, and that equilibrium we're affecting. And it could change in a way where there's a new equilibrium, which does not support us as well. And,
yeah, we didn't talk, David, at all about the plastics.
Right.
When we sailed out of Panama and up to up the coast, there's a place where 3 major ocean currents come together, and all you see out there in the middle of the ocean for miles is plastic trash and other kinds of trash. We actually sailed close to a refrigerator that was floating in this miles wide plastic and garbage collection in the ocean. And one of the crew members wanted to go open it and see if there was beer in there. But it's actually, we find plastics now everywhere. So I'm sure many of you have been to Lake Tahoe. It's one of the most pristine looking bodies of water and that we have for an inland lake, and it's truly beautiful. We took a plankton net up there and just towed it behind my antique power boat for an hour, about 10 feet deep, and collected. The net got filled with microplastic. So it doesn't show up when you just look in the water. But here's this most pristine lake that for all practical purposes you think is pure drinking water. And it's full of degraded plastic down to very small pieces that fish and people consume.
That actually is a perfect segue into the next question. And I think it's the last one we have time for. So I'm sorry, everyone. I wish we could do this for an hour, but Steve Sherman, who actually works on microplastic issues, he's got a plastic solution to try to fix that, was asking, Steve, can you kind of explain that question a little bit about the ocean fighting back?
Well, we know that there's a lot going on in the ocean that we don't know about. And I appreciate what Dr. Venter is doing and helping us discover and explore. But if we talk about this sequestration of gases, and this gas exchange is created at the very top of the oceans, and we mess with the equilibrium, maybe the equilibrium will mess back with us in ways that we don't know, or ways that we don't understand. And this could happen on a very large scale. I didn't know if Dr. Venter had any insights into that where floating plastics would interfere that gas exchange. That's I think a possibility for severe problems we just don't know a lot about. Not a lot of people are talking about that.
Well, we're actually finding on some of these microplastics in the ocean, there's microbes on the surface that is learning to digest some of that. The negative side of that is, it's converting it into chemicals that wouldn't normally exist in the ocean. So, we don't know what the effect is going to be on the large scale that we have. And so the experiments that some want to do with geoengineering, messing with the system that produces the oxygen that allows us all to live.
Maybe not such a good idea.
It is not the brightest idea, particularly with still our fairly naive understanding of what's out there in the ocean chemically.
Thank you.
All right. Well, thank you so much. I really appreciate your time. I know we all do. That was definitely a fascinating panel. Thank you, David. Well executed moderation there. I think we all learned a lot today. And hopefully, Craig, we'll have you for a little bit at least. I don't know how busy you are today, but I'm guessing it's busy. So I know there are a lot of really good questions that were coming through in the chat at the end there. And maybe you can message Craig on the platform and see if he is available to answer some of them.